The overall goal of this experimental strategy is to investigate the functionality of disease-associated non-coding variants.
This method can help answer key questions in genomics, such as how genetic variants that don't actually change amino acid sequence still contribute to disease phenotype.
The main advantage of this technique is that it provides a streamlined process for assessing genetic variants for functional activity and gives insight into regulatory proteins and pathways affected.
This technique can benefit the investigation of any disease with a candidate causal variant because the procedure for analyzing these variants is universal regardless of the phenotype being studied.
Though this method can provide insight into human disease, it can also be applied to mutations in any organism.
Nuclear lysates will be prepared from B lymphoblastoid cells in this experiment. However, this procedure can be applied to most cell lines. After counting cells using a hemocytometer, spin down the desired number of cells for lysing. Aspirate the media, add 10 milliliters of ice-cold PBS, and spin down the cells again. Wash the cells a second time with PBS, and remove the PBS after the spin. Resuspend the cell pellet in ice-cold PBS using 1 milliliter of PBS per 10 to the 7th cells. Aliquot 1 milliliter of the cell suspension to 1.5 milliliter microcentrifuge tubes so that each tube contains 10 to the 7th cells in PBS. Centrifuge for 2 minutes and aspirate off the PBS. Add to a working stock of CE buffer 1 millimolar